All right, good afternoon. Glad to be here. Today I'd like to talk about how focused ultrasound has become and gaining momentum as a new modality of brain stimulation. And I'd like to share our experience in studies through small animals to humans. So why do we need non-invasive brain neuromodulation? Well, pharmacological approaches are not region-specific, and region-specific neuromodulation techniques may provide unprecedented opportunities in neurotherapeutics and more. For example, modification and the control of regional brain function maybe offer the tools to assess the brain function. Probably it's a new way of doing brain mapping. So there are several techniques indeed out there to do the neuromodulation. ECT, DBS, ECS, somehow they are all invasive. There are a couple of non-invasive options out there, which is TDCS, TACS, and TMS. But these methods, the area of modulation is still quite large, and they cannot reach deep brain areas. So we want both spatial selectivity and deep brain penetration, and we believe FOSS can provide a unique solution. So I personally believe we are in the middle of Renaissance period for therapeutic ultrasound, and we owe a lot of it to the development of a transcranial FOSS system. Typically, we use a low frequency range, which is typically less than 1 megahertz for better scope penetration. And breakthrough was made in the development of a multi-array FOSS transducer that consists of multiple arrays of small ultrasound transducers, thousands of them, hundreds of them, and we can independently control their activation to correct for the phase aberration caused by the skull. And this helmet-like device that you can see from the corner on the left it can consist of multiple array of transducers, and it is used for thermal ablation of brain tissues when it operates in the high intensity range. On the other hand, you can utilize a single element transducer that may use an acoustic lens or just by shaping its transducer surface in little curvature so that it can zoom in the acoustic pressure wave into a specific brain area. So multi-array transducers, they offers advantages over the single element by delivering very tighter focus into the deep brain area. But also single element, it can probably do a lot of things altogether. Multi-array transducer unit, it's a little bit expensive and pricey, but it also requires a surgical grade patient preparation. For instance, it requires a shaving of the hair, as well as you have to put a skull screw. So it may not be that applicable for routine use in outpatient clinical setting. So one can see after a hybrid approach, which may take the advantages of the two. So how can you do the neuromodulation using focused ultrasound? We apply low intensity FOSS as a train of pulses with sufficient interpulse intervals. And non-thermal mechanical pressure waves are given under image guidance to visualize invisible acoustic focus. We found that either stimulatory or suppressive effect can be inducted using different pulsing schemes. Although the jury, we cannot really get the definite the parameters for doing it, but we found the stimulatory effect can be anticipated using high duty cycle and short burst sonication duration compared to a suppressive counterpart. And also we should use higher acoustic intensity to achieve the stimulation than the suppressive ones. So let's take a look at some of our earlier experience in animal. This is the rabbit that are lying over the bed, and the sonication is giving from the bottom to top through the water bath that are coupling the transducer to the skull surface. And by depending on adjusting the acoustic intensity or the location of the sonication, you can actually modulate the degree of movement in rabbits. Okay, and we also delivered the sonication to rats on the right-hand corner. We deliver the sonication, stimulatory sonications to M1 area that are responsible for stimulating the tail or the forelimb. In this case, it is ipsilateral, contralateral part, and you can see the muscle movement can be elicited. So after playing with the stimulatory effect, we shrunk down the transducer a little further. We actually made it into a small little size of the finger, and it weighs only 9 grams, so that rat can wear it like a helmet. And we delivered sonication through little applicators that are 3D printed to tailor-make their sonication focus in the skull. And somehow it didn't really start, but can you start the video from there? Oh, there we go. Thank you. So we delivered the sonication to, well, it's frozen again. Sorry about that. We can stimulate the sonication, stimulate the M1 area in both awake and anesthetized state, and it can elicit a corresponding movement from the rats. So let's change gear and take a look at the suppressive effect of FUS on the CNS. After narrowing down the parameters that suppress the regional brain activity, we set out to test if the FUS can suppress the epileptic activity. So under the guidance of EEG, acute stage epilepsy was induced by injecting PTZ, and suppressive FUS sonication was delivered to the thalamus. And we monitor that the EEG can be, ictal EEG can be suppressed, and also with the stop of a seizure activity. And this is the video of what we have seen. So this rat just got an injection of PTZ. PTZ is a GABA inhibitor. So if you give GABA inhibitor, then there is over-excitability in entire brain. So soon it developed a seizure, just like that. And if you do not do anything, the seizure activity lasts about good 1 hour. So, and it's very potent. So there is not really an easier way to stop it. So we delivered the sonication on about 1 minute later, which will start very soon. We delivered the suppressive sonication for about 30 seconds or so to the thalamus. Why thalamus? Because it is the main circuitry of entire brain. So we decide to shut down the main circuit in a way. And sonication was delivered about 30 to 1 minute. And then immediately, we didn't really see any effect, but in about 10 seconds or so, we start to see the stop in seizure activity, as well as EEG ictal signatures. Right? So after that, we decided to see if the sonication have any effect on the modulation of neurotransmitter activity. So while we are giving sonication to thalamus, we measured the GABA and glutamate, dopamine, and serotonin level from extracellular space from the rat frontal lobe. And we saw the glutamate level didn't change, which is good news because there's no glutamate toxicity associated with it. GABA level went down, whereas dopamine and serotonin level went up. We do not know exact mechanism behind this phenomena, but we found that this result suggests a very good promising result that may have a treatment for the neurological or psychiatric disorder that may have something to do with aberrant neurotransmission. Let's change another gear to the effect of FUS on PNS. Let's look at the effect on myelinated nerve bundle. We found the FUS can disrupt nerve conduction in earthworm model on the left, and it temporarily reduces the nerve conduction peak temporarily, but it didn't affect the nerve conduction velocity. So motivated by that, we delivered a stimulatory FUS to the abducens nerve in rats, ipsilaterally. As you know, abducens nerve, that they abduct the eyeball to the sideway. And let's see what actually happened. When you turn on the sonication, the eye rolls backward, only one side, at the same side of the sonication itself. And if you turn it off, and eyes rolls backward into its neutral position. So we can actually show this one over and over again, but for the sake of time, let's move on to the next slide. So after testing out this idea in small animals, we decide to try this technique in larger animals. In this case, it's sheep. We deliver the sonications to SM1 and V1, and we measured the EMG activity as well as evoked potential. And we found that actually it was done under the image guidance to accurately deliver acoustic focus to the M1 and V1 area in sheep. And we found a certain minimum acoustic intensity is needed for the stimulation. And the responses increases with a acoustic intensity, which is actually similar findings that Elisa Konofago has found. And with a collaboration with a Korean Primate Research Center, we applied the stimulatory fuss to the M1 in monkeys. And see cute little hand move according to the stimulation. So they used a small but multi-array transducer that have a beam steering capability. So they're optimizing their parameters. So let's see how they improve or continue to generate the similar results over time. So motivated by these results, we also applied the focus ultrasounds to humans. In this case, the transducer was two transducers were actually mounted to an applicator. They are mounted to the helmet, bike helmet in this case, and we delivered a sonications to S1 and S2 of the brain simultaneously and independently. And under the image guidance, we used individual specific fMRI and CT and anatomical MRI to navigate the acoustic focus for that specific individual. And we successfully elicited a tactile sensations according to the simulation. It was very interesting to see this activation of S2 alone was able to generate the tactile responses. And then we decide to move on to see if the FUS can stimulate V1. So now the transducer was located inside of the MRI coil. It was done on the 3 Tesla. And the stimulatory ultrasound was delivered to the V1 under the image guidance. We know the geometric relationship with respect to the head anatomy based on the negative imprint that are seen on the T1 image. And the sonication elicited not only the phosphine, and we also measured the fMRI during the sonication using event-related design. And we found it not only elicited the activation from the V1, but also activated the much of other cortical areas, which include frontal lobe as well as several limbic area, which suggests that the sonication can elicit more activation that are related to high-order cognitive processing. So what would be the potential application? Obviously, you can expect various neurotherapeutic encompassing various neurological and psychiatric disorders. But it can be used as a cool research tool. For example, we apply the technique to study brain-to-brain interface. So brain-to-brain interface is like this. So EEG-based brain-to-computer interface was used to detect a hand motor brain activity from one individual. And then FUS technique is used to can stimulate the somatosensory area of the another person and was used as a computer-to-brain interface. So what do we have in the middle? A computer is already widely connected through the internet, so we can establish a BBI. And let's see how it was implemented. So we divided a pair of subjects, human participant, into 2. One is a sender on the left, and one is a receiver on the right. And they are separated about 20 miles away in geometrical region. And the sender was performing a motor imagery hand task of the right or the left hand, depending on the target that appear on the screen. And by detecting the mu rhythm, EEG mu rhythm that are associated with the imagery, you can translate that intention into a computer command. And the computer command can be transmitted to another part of the place that actually activate the left or right area of the SM1 to stimulate the brain area. And upon the receiving the stimulation, the receiver tapped their right or left finger to acknowledge the site of stimulation. So remote hemispheric link of the brain function, although it is quite rudimentary in this case, can be established between two individuals. So as a concluding remark, what would be the potential mechanism? Well, we know it is not temperature related. We didn't really see any temperature changes through MR thermometry. And it is not electromagnetic. It is observed without MR environment. So the eddy current, those things are not included. So therefore, it's most likely through mechanical effect, and it is mediated by the mechanoreceptor or ion channel activity. So there are a couple of hypotheses out there, probably 5 or 6 out there, and I listed a few of them. First one is bilayer sonophore model. It models the downstream effect from the changes in membrane capacitance changes that are induced by the ultrasound. And it is could be possibly mediated by the activation of astroglial system through calcium signaling. And it could be elicited by stimulation of group or ensembles of inhibitory versus excitatory neurons, for example, interneurons versus pyramidal cells. And lastly, it could be modulated by the neurotransmitter activity, or it could be combination of any of these potential mechanisms as well. As we are looking into just the tip of the iceberg in this neuromodulation, we need more studies to reveal its true utility in healthcare. And I would like to acknowledge my collaborators and funding sources. Thank you very much.